Good morning students. Today we are going to continue our topic, that is recombinant DNA. So this topic, that is recombinant DNA, that is from paper 2 PS EBS 402, and under the paper 2 we can find out 4 units, out of which the first unit, that is environmental biotechnology. So under environmental biotechnology we can find out the topic, that is subtopic, that is recombinant DNA technology. So yesterday I have already sent you a lecture regarding the tools in the rDNA technology, recombinant DNA technology. So today we are going to see the topic, that is recombinant DNA technology. Recombinant means recombining the DNA. From the name itself, it is very clear that recombinant means a recombining of the DNA. So it is one of the this topic comes under the genetic engineering, and it is one of the applied branches of biotechnology. So you can say that we are studying somewhat related to the environmental biotechnology, so we must know what is genetic engineering. So how can we define the genetic genetic engineering? It is one of the applied branches of the biotechnology. So here in the picture we can find out interested gene. This is the blue gene. So interested gene is digested with the restriction enzyme, and this target gene is then joined with the another DNA fragment. So the interested gene is first isolated, then it is cultured, and then it is introduced into the vector. In this way we are going to apply this biotechnology techniques. So definition of the genetic genetic engineering is the addition. So here we are going to add, we are going to remove, we are going to replace, or we are going to repair a part of the genetic material resulting into the change of the phenotype of organism is called genetic engineering. मतलब एक ऐसा प्रकार का प्रोसेस है जिसमें कि हम या तो ऐड करते हैं, या फिर रिमूव करते हैं, या फिर रिप्लेस करते हैं, या फिर रिपेयर करते हैं कौन सा जेनेटिक मटेरियल पार्ट। और उसके कारण क्या होता है, जब हम रिप्लेस करते हैं या फिर ऐड करते हैं या फिर रिमूव करते हैं या फिर रिपेयर करते हैं तो एक चेंज आता है, दैट इज द चेंज इन द फिनोटाइप, बिकॉज वी आर जस्ट रिप्लेसिंग और रिमूविंग द जेनेटिक मटेरियल। हम वो जेनेटिक मटेरियल क्या करते हैं, इंटरेस्टेड जीन निकालने के बाद उसे ऐड करते हैं। इसके कारण क्या होता है, देयर इज ए चेंज ऑफ फिनोटाइप ऑफ ऑर्गेनिज्म के फिनोटाइप में जो दिखावट में चेंज आता है, एंड दिस इज कॉल्ड द जेनेटिक इंजीनियरिंग. So this is somewhat related to the definition of the genetic engineering. So genetic engineering is also called as gene manipulation, or it is also called as the recombinant DNA. So we can say that genetic engineering can be also called as the recombinant DNA. Recombinant DNA. And all the activities in genetic engineering are done very carefully. So these activities are done very carefully under highly controlled laboratory conditions. मतलब एक लेबोरेटरी कंट्रोल कंडीशन में ये सारे प्रोसेस कैरी आउट किया जाता है. द फील्ड ऑफ जेनेटिक इंजीनियरिंग इज वेरी वास्ट, इट हैज़ गॉट अ वेरी वास्ट स्कोप, एंड यू कैन से and got the tremendous variety of application. आज के now in this decades we can say that कि it has got a variety of application. बहुत सारे application होने के कारण this is to be normally practiced. So today we are going to see the tools. So yesterday I have already sent you the tools used in the recombinant DNA. So I hope all of you have gone through it. So what are the different tools? So in order to cut, certain enzymes are used, and these enzymes are said to be the restriction endonuclease. So these enzymes, they normally act as a scissor. So you have seen the knives or the scissors. So this restriction endonuclease or the enzymes. So the tools which are used here are as follows. Like first one, that is said to be enzymes. So it requires use of different type of enzymes, and these enzymes may be the restriction endonuclease हो सकता है. It may be the DNA ligases. Ligases, from the name itself, a ligase means to join. So restriction enzymes or endonuclease, these are the enzyme which is used to cut. So you have seen the knives or you have seen the scissor. So in case of clothes also, we require to first cut it, then we have to join a new piece. Similarly here also, instead of using the scissor, we are using an enzymes as a scissor or knives. So restriction endonuclease, they are the normally they are used to cut the DNA at a specific point. So if the DNA is present, we have to cut the DNA at a specific point, then we require certain enzymes, and these enzymes are said to be the restriction endonuclease, and they are called the biological scissors. Biological scissors or molecular scissors or the chemical scalpels or the chemical knives. So restriction enzymes, they are said to be the knives, chemical knives or biological scissors, and they are used to cut the DNA at a specific point. हमें जो interested gene है उस साइड को हमें कट करना पड़ेगा, and for this we require the restriction endonuclease. So restriction endonuclease may be of restriction enzymes may be of three types. It may be restriction enzymes 1, restriction 2, and restriction enzyme 3. They are the different type of restriction enzymes, and they are having a particular function. According to that function, they are utilized. Next, coming up to the vector. So it is a carrier DNA. What is a vector? Vector is a carrier DNA molecules to which the fragment of desired DNA is attached. So if it is a carrier molecules and this is our interested part of the DNA, so if we are going to join this DNA, so consider this is the interested part of the DNA, so we are going to join it. So we can say that this is the vector. So it is a carrier DNA which carries the interested gene. So it is a carrier DNA molecules to which the fragment of the desired DNA are attached and carry to require and carries to required site. आपल्याला आपको जहाँ लेके जाना है वहाँ पे उस साइट पे हम इसे लेके जा सकते हैं. And it may be the plasmid or cosmid. So this vector may be the plasmid. It may be the cosmid DNA, or it may be the various type of plant and animal viruses, and sometimes may be the bacteriophages. So bacteriophages DNA, plasmid and cosmid, these are normally the vectors normally which are used in order to insert our interested DNA, and then they are cloned. And some commonly used vector plasmids are these plasmids maybe are PBR322. These are certain plasmid vector. One more example, that is PUC19. This is again a vector, plasmid vector है. And some virus based vector plasmids are bacteriophages, like maybe bacteriophages where you can find out the lambda, LAMDA. So lambda phage, bacteriophage, and certain example of the cosmid vectors may be the YAC. So these are the few examples of the vectors, that is plasmid vector. We can say that PBR322 and PUC19. These are the plasmid vector. Cosmid vectors may be YAC. Normally शटल वेक्टर बोलते हैं हम. ईस्ट आर्टिफिशियल क्रोमोसोम्स, हम YAC stands for ईस्ट artificial chromosomes, normally extracted from the yeast. And next is the bacteriophages, which includes the lambda vectors. So you must know what are the different type of vectors, and now which are the different type of vectors which are used in order to insert the interested gene inside the vector. And what do you mean by the target DNA? The target DNA is the desired fragment of DNA, the interested DNA, part of the DNA, which are to be transferred from one organism to the another by using some vector is called the target DNA. So what do you mean by target DNA? It is the desired fragment of DNA which is to be transferred, जो कि हम ट्रांसफर करते हैं from one organism to the another organism by using the vector. वेक्टर को यूज़ करने के बाद हम क्या करते हैं, हमारा इंटरेस्टेड जीन हम ट्रांसफ़र करते हैं या फिर उसका कॉपी करके लेते हैं. दे आर सेड टू बी द टारगेट डीएनए. Next, coming up to the steps which are involved in the genetic engineering or recombinant DNA technology. So what are the steps which are involved in the recombinant DNA technology? This RDT, it is a short form of recombinant DNA technology. So the process which involves the following step, out of which the first step is the isolation of the desired gene. First step is the isolation of the desired gene. Here the cell or the organism from which the required gene is taken is called the donor. So जिस organism से हम required gene लेते हैं, they are said to be the donor. DONOR. And from the DNA of the donor, a particular gene is selected and isolated by using the restriction endonuclease enzymes. So the interested part of the gene is selected and then they are cut by means of the restriction enzymes. And the donor DNA containing the desired gene is called the passenger DNA. So if the donor DNA containing the desired gene, हमें जो desired gene चाहिए है वो इसको हम क्या बोलेंगे, passenger DNA. So they are said to be the donor, and which are cut by means of the restriction enzymes, they are also said to be passenger DNA. Next is the selection of the vector. So a suitable vector, that is gene carrier, is now selected. क्योंकि हमें क्या करना है, उस interested gene को दूसरे vector में डालना है in order to bring about a particular function. एक living cells में डालना पड़ेगा, नहीं तो हम उसे कट करके डायरेक्टली बाद में डायरेक्टली जहाँ चाहिए वहाँ पे इम्प्लांट नहीं कर सकते हैं. क्यों? क्योंकि वो फंक्शन नहीं करेगा. सो इन ऑर्डर टू फाइंड आउट वेदर इट इज़ फंक्शनिंग और वर्किंग, इट शुड बी इम्प्लांटेड इनसाइड अ organism. So a particular vector is selected. So a suitable vector, that is gene carrier, हम क्या उसे बोलते हैं, वेक्टर को जीन कैरियर, क्योंकि वो इंटरेस्टेड जीन कैरी करेगा. सो जीन carrier is now selected, which may be generally a plasmid हो सकता है, phages हो सकता है, cosmid हो सकता है. Already discussed about it. Next is the formation of recombinant DNA. How does the formation of recombinant DNA takes place? So with the help of restriction enzyme, द वेक्टर डीएनए इज कट इनटू स्पेसिफिक पॉइंट्स। हम क्या करते हैं, एक ऑर्गेनिज्म का ये जो पॉइंट है, स्पेसिफिक पॉइंट पे हम रिस्ट्रिक्शन एंजाइम से हम क्या करते हैं, कट कर लेते हैं। एंड द कट एंड ऑफ वेक्टर्स आर sticky and cohesive, and the selected and isolated desired gene is now inserted into the vector DNA। इसको हमने कट कर लिया, उसके बाद हमने क्या किया, एक प्लास्मिड लिया, so this is the consider, this is the desired gene which we want। This is the vector plasmid। Now what we are going to do is that we are going to replace this part over here। So here again we have to cut this part। So we have cutted the desired part by means of restriction enzyme. Now we have inserted inside the vector। Now we got the particular vector with the interested gene। Now they interested into the vector DNA which is made to fuse there with the help of ligase। So interested gene is fused with the vector gene with the help of, हमें चिपकाने के लिए कुछ तो चाहिए होगा ना। So in order to attach we used to make use of certain enzymes again, and these enzymes are said to be the ligase। So DNA ligase, which are normally used in order to stick the ends। So here enzymes will come and attach the ends of the interested DNA with the vector gene or the DNA। Now the vector DNA with a new introduced gene is called the, this is said to be the recombinant DNA। Why? Because we have altered the DNA, that's why it is said to be the recombining। We have combined the DNA once again, that's why they are said to be recombinant DNA। Next steps in the recombinant DNA trans technology is the gene transfer to the host। So this plasmid is now introduced into a suitable bacterium। हम क्या करते हैं, इस plasmid को कहाँ introduce करते हैं, inside the bacterium। And so this is introduced inside the bacterium, and now this bacterium may be E. coli, it may be the E. coli for the cloning। So after the cloning you can say that it may be the Bacillus subtilis and the Saccharomyces cerevisiae, they can be used as a host, and the host should be non-pathogenic and should be harmless microorganisms which is easy for cultivation। So that's why they are using normally the Bacillus, Bacillus, Bacillus subtilis or the Saccharomyces cerevisiae, 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 AE। So normally these as a host they are used in order to clone the particular recombinant DNA, and the host should be, ये जो होस्ट होना चाहिए एडर में भी दी कोलाई हो सकता है, बैसिलस हो सकता है, सैकरोमाइसिस हो सकता है। तो दिस शुड बी हार्मलेस होना चाहिए, पैथोजेनिक नहीं होना चाहिए, एंड इजी टू कल्टिवेशन, हम इजीली उसे कल्टिवेट कर सकते हैं। सच अ बैक्टीरियल सेल विद such a recombinant DNA is called the transformed host। So if you can find out a E. coli or the Saccharomyces cerevisiae with a recombinant DNA, इस recombinant DNA के साथ अगर है तो इसे हम क्या बोलेंगे, transformed host, transformed host, because we have transformed the host, and the incorporated our DNA into the bacterial cell is generally done by means of electroporation, by means of electroporation. So last step, that is said to be cloning. This is the transformed bacterial cell and is now allowed to grow on a nutrient medium. यहाँ पे हम क्या करते हैं, जो transformed DNA recombinant DNA है बैक्टीरिया के अंदर में, उसे हम allow करते हैं in order to grow. हम क्या करते हैं, we used to provide almost all the condition or nutrient medium where they can multiply or grow, and so it results in the formation of large number of transformed bacterial cells. So we get a large number of transformed bacterial cell, and all these cells have a copy of recombinant DNA. So all these cells, they are having the recombinant DNA. These are the steps which are involved with the recombinant DNA technology. So today we have seen what are the different steps. First is isolation of desired genes, second is selection of vector, third is the formation of recombinant DNA, fourth is a gene transfer to the host, and fifth one is the cloning. So we have seen what is target DNA, we have seen what are the different type of vectors which are used in the cloning. Next, what are restriction endonuclease, what is the function of endonuclease. Next, what are the different type of tools which are used in the restriction, or different tools which are used in the recombinant DNA technology, and what is mean by the genetic engineering. So these are the different aspects which we have seen inside the recombinant DNA technology. So see you tomorrow. Thank you for watching.